Then next one. Engineering of metabolic pathways in plants can be achieved by introduction and overexpression of appropriate candidate genes using transgenic technology. You all know that through transgenic technology, you can genetically modify the metabolic pathways either through introduction of a gene or through overexpression of a particular gene. Now the figure given below represents a biochemical pathway in plants where a precursor molecule A is converted into its products T and X through a series of enzymatic reactions. So A is converted to T and X through series of enzymatic reactions. Enzyme 1 to 5, 1, 2, 3, 4 and 5 are involved in this pathway. Okay. Scientists attempted to increase the level of X by introducing an additional copy of the gene for enzyme 5 under transcriptional control of a strong constitutive promoter. So what did the scientists do? They attempted to increase the level of X. They want to increase the level of product X by introducing additional copy of the gene for enzyme 5. So enzyme 5, they have introduced an overexpressed under transcriptional control of a strong constitutive promoter. Okay, so they have overexpressed enzyme 5. However, the developed transgenic plant did not display a proportionate increase in the level of X. Now, why did the plant did not display an increase in the level of X? The following statements were proposed for explaining the above results. Enzyme 4 has greater affinity for D than enzyme 3. That may be one of the reasons. Why? Because enzyme 4 has greater affinity for D, so it will convert more amount of D into T and little amount will be converted into E by the enzyme 3 because enzyme 4 has greater affinity for D than 3. So it will be converted, it will convert D to T instead of E. That may be one reason. So, and also what will happen as a result of this is that it will limit the availability of enzyme of product E to enzyme 5 to convert into X. That is the outcome when enzyme 4 has greater affinity for D. The next one. Feedback inhibition of enzyme 5 by compound X. That also may be one of the reason why you got the lesser level of or lesser amount of or lesser concentration of X. Substrate limitation for enzyme 5. That I told you already when I explained enzyme 4 had greater affinity for D than for enzyme 3. So the answer here is A, B and C. Option 4.